Okay, so let's get started. So welcome everyone to today's Zoom seminar. And then thanks a lot everyone to come jump on the Zoom during the weekend, and in particular thanks to our speaker. So today it's really my pleasure to welcome today's speaker, Dr. Stanley Huang from Ohio State University. Stanley actually been a friend for a while, and then he just moved from Case Western to Ohio State University. So I think before the seminar, I just want to give a shout out for Stanley. He just started the new lab in OSU, and then I think he's doing really cool immune metabolism research. If anyone had interested, I think you should definitely check out his website and check out his study. You might find it interesting. So Stanley currently is associate professor in microbiome infection and immunity in Ohio State University College of Medicine. So he actually complete his undergrad study in the Chang'an Christian University in Taiwan, and then continue his training in microbiology in Chang'an University, also in Taiwan. So from 2006, Stanley started his PhD training in Imperial College London, England, in molecular and cellular biology. And then afterwards, he continued his postdoc training in Department of Pathology and Immunology in Washington University School of Medicine. And then so he first studied his postdoc training in Turin Institute with Edward Pierce, and then I think the lab actually relocated into in Washington University. So he moved to Washington University, continued his postdoc training in Department of Pathology and Immunology. So from 2018, Stanley started his independent research program as assistant professor of immunology in Department of Pathology in Case Western University. And then just this year, he relocated from his lab from Case Western to Ohio State University. Researching Stanley's lab mainly focuses on understanding the interplay between the cellular signaling and the metabolic regulation, which dictate immune cell activation during inflammation. The main goal of their work are to determine the specific environment cues and nutrition changes and unusual metabolites which can control the metabolomic and epigenetic programs to selectively impact innate immunity in inflammatory diseases and cancer. His lab actually mainly focuses in several different aspects, which including metabolic reprogramming in macrophage fate and immunity, which I think he's going to talk about today. His lab trying to understand the metabolic and then bioenergetic requirement for dictating the cellular function of macrophage, and then to try to develop tangible clinical immune therapies which targeting macrophage through metabolism for cancer and infections. And the second part focus of his lab is metabolic intervention of ILC3 in GI inflammation, which they try to understand the molecular mechanism such as mTOR pathway that regulates ILC3 development and immunity during intestinal inflammation. And eventually, they try to their work may assist to identify new therapeutic targets, and then based on that, which actually can metabolically manipulate ILC, hopefully in human disease such as IBD and colon cancer. Stanley actually really did a very productive postdoc period, and then also during his tenure track research. And based on these achievements, he received numerous awards and honors. I think I'm not gonna list them all, but just selectively, 2019 he got Cancer Research Institute Investigator Award. 2020, which he got Velo Sano Bike to Cure Pilot Awards 2022. He got Melanoma Research Foundation Career Development Award, and 2023, this year, he got American Cancer Society Research Scholar Grant Award. So I think without further ado, Stanley, we are really excited to look forward to your talk. Please proceed.
Thanks, Tran, for your nice introduction. And it's my great pleasure to be here to share our study with you guys. And as well, Tran say, I just relocated from CASE to OSU, and then today I'm going to present the study we did in the CASE for a couple of years, and the leading author is my PhD students. So today I'm focusing on the macrophage metabolism in the tumor microenvironment. So we know that immunology is often about interactions, and then we know when you have a pathogen insult or infections or tumor invade, you will initiate the tumor's immune system to have an immune activation, and the immune system will try to kill or to expel these pathogens to protect the host. Meanwhile, this interaction not only between pathogen and immune system, the pathogens or tumors also cause the stress response to the tissues. So these variance factors will initiate some stress response, cause damages to the tissues. And these will lead to the tissue dysregulation. And with the severe dysregulation, it will cause the host death. And meanwhile, the stress damage also will cause the immune system responses. And this response will try to eliminate or reduce the stress from this pathogen insult. So this stress damage response will cause immune system have a immune responses to do the tissue regeneration. And this regeneration will try to control the damage, prevent hosts from death to survive, to survive. And this is very complicated interaction, not just between the pathogen and the immune system, but also tissue and the immune system. And then we know that during this process, normally it's a metabolic exhaustion. However, on the other hand, immune cell system under this situation, they try to adapt the metabolism, try to maintain their homeostasis to balance this infection or immune system response. So this is often about interaction between immune system and pathogens, and then this also involved in the metabolism. And we study metabolism. So metabolism basically is about transformation, not interaction, so transformation. So metabolism can comprise catabolism or anabolism from one metabolite to another metabolite. So before this, and you have more food, you become this. So it's about transformation from one thing to the other thing. So it has been appreciated these years, showing that metabolism is required to support immunological functions or immune system development or effective function. And then this metabolic reprogramming match the immunological functions, like, for instance, doing the inflammation M1 macrophages or pro-inflammatory macrophages, they highly depend on glycolysis. They use glycolysis to generate ATP, and they have a low mitochondrial function. Then they will try to respond to the environmental challenge. On the other hand, anti-inflammatory M2 or Treg or memory CD8 T cell, they mainly rely on mitochondrial function or fatty acid oxidation. And they use this metabolism to do the surveillance or suppression function in the body. So it's very well known that metabolism definitely can control or dictate immune cell function in a different aspect. And our lab is one of our interest cell is macrophages, and we know macrophages are important frontier immune cell in our body to fight against pathogens or tumors. And roughly speaking, macrophages can divide into two subsets, roughly speaking. So one is pro-inflammatory macrophages, people call it M1 macrophages. So these macrophages play a role in atherosclerosis, diabetes, or bacterial infection, virus infection. On the other hand, the macrophages have an anti-inflammatory phenotype or immunosuppressive macrophages, or people always call it M2 macrophages. These macrophages play a role in helminth worm infection, tumor microenvironment, or wound healing, and also the fibrosis. So these two types of macrophages, they play different role and have different functionality in the different environment to fight against the diseases. And our lab particularly interested in tumor microenvironment, in tumor microenvironment and studying M2 or anti-inflammatory macrophages. And we also can use the worm infection to mimic this phenotype. And we know that in during the infection, during the cell in response, when you have a pathogen, virus, bacteria, or cancer, worm insult, these macrophages will have different responses. So the first wave is you will cause the host defense inflammation, the phagocytosis. And second wave, this cell will try to proliferate to maintain the tissue homeostasis, and then make sure cell is normal and fight against the pathogens. Later on, you have the third wave called clearance, resolution, repair, or differentiation, try to make sure host or tissue can eliminate this pathogen or inflammation. And during this process, we're interested in metabolism changes. So we try to understand what the cell signaling or metabolic requirement for this process, what metabolite or metabolic pathway involved in these different stages of the infection or diseases. So we, as I say, we interested in macrophages and particularly in the tumor microenvironment. So we took the data from the previous published data showing that in the most solid tumors, about 30 to 50% of infiltrated immune cells are macrophages or monocytes, and they play an important role in these tumors, and then these cause the immunosuppressive environment. So what we can do to change this situation, make this immunosuppressive environment less, and then make them more like anti-tumor immunity. So we studied metabolism. We know tumor microenvironment is very complicated, and then the environment is very harsh. We know that in the normal tissues, you have normal oxygen, normal nutrient like glutamine, and very low this waste lactate, and then have normal immune infiltration. But in the tumor microenvironment, tumors secrete different metabolites or consume huge amount of glucose or metabolite that those are essential for T cell, for instance. They maintain T cell immunity. But in the tumor microenvironment, this situation make T cell impaired. And then this situation, like it has been shown that from the Ruslan multitope, they show tumor microenvironment secrete lots of lactate, the prime macrophages become immunosuppressive M2 phenotype, and that will promote tumor progression. And then they use tons of arginase to consume this environment nutrients and impair T cell function. So we want to know what else that will dictate these tumor social macrophages, or why tumor social macrophages can survive or do so well in the tumor microenvironment, not like T cell. T cell in the tumor microenvironment is definitely impaired or non-functional to fight against tumor. And then tumor social macrophages, they are fine and they can do well. So we want to understand what makes them become so powerful to promote tumor progression. So we took a data set, a publicly available data set from lung cancer patient samples and then liver cancer patient samples, looking at the tumor social macrophages versus the non-tumor social macrophages. We found that in the lung cancer patient, tumor social macrophages are enriched with mitochondrial phosphorus and also glycolysis, in contrast to the T cell. And then similarly, in the liver cancer patient, tumor social macrophage also enriched with mitochondrial function and glycolysis. So why is this? So why these macrophages have intact metabolism to control the immunosuppressive property to promote tumor progression or tumor immune evasion. So we want to understand what is the detail, what control this situation. And then we know that the current situation to kill tumor or treat tumor is immune checkpoint blockade. So the PD-L1 or PD-1 blockade has been well applied to the clinic. However, only 10 to 30% patient benefit from this treatment. So the major roadblock of this environment itself is passed through this pro-tumoral or immunosuppressive M2 macrophages. So these M2 macrophages always cause the immune checkpoint blockade resistant or treatment resistant or reoccurring also due to this immunosuppressive M2 macrophages. Can we do something to eliminate or decrease this phenotype and make this immune checkpoint blockade even better? So our central questions are, is there any metabolic checkpoint that can dictate or influence metabolic adaptation or pathway that is important for macrophages in the tumor microenvironment? And second question is, can we edit or manipulate this process in macrophages that will promote or inhibit the effector function? So today I'm going to tell one story, and I divided two parts. And this story already published last year in Nature Immunology. So the first part we will show you about how the cellular stress response in these tumor-associated macrophages. Second part is about how this stress response control this intrinsic serine non-essential amino acid metabolism that support immunosuppressive macrophages in the tumor microenvironment. So before that, I want to simply introduce you about what is stress response in our body. So normally when you have acute or chronic stress, it's normally associated with increased inflammation activity. So give you an example I found very interesting in published in BBRC in 2013. So this study, they used two groups of mice. One is controlled without any influence, and the other group is these mice suffer from chronic screen stress. Imagine if you have a small kid and the kid screaming all the time around you, what will happen? So these two groups of mice can see they found this group of mice with chronic screen stress. Their body weight decreased, and surprisingly, the chronic sound stress causes a shift from Th1 to Th2 response. So definitely, the environmental stress challenge will affect immune responses, and then of course they will affect metabolism. So which one happened first is not clear. So I found the paper interesting. So when you under stress, your metabolism will be altered. And this change may affect immune responses. It's like very interesting interaction. So we know stress response is affecting immune function. And inflammation impose stress signal to affect the energetic demands and also fitness and of the immune cells. So we know that the stress response in cell had the process called ER stress response. So ER stress response, it can overcome any kind of stress from the environment or intrinsically to maintain the cell homeostasis. We know that in our body, you have normal adaptive response, normal stress, very normal, you adapt. So the stress is very low. But when you have chronic disease such as diabetes, obesity, your body will slowly increase the chronic stress, and this will lead to loss of metabolic control. So you have a huge body weight gain from the obesity, something. Furthermore, you have acute stress challenge, like all of a sudden you drink huge amount of glucose, they will cause toxicity. So this we will have a cell cytotoxicity that immediately cause a cell death or apoptosis. So stress response can handle this situation in our body or in the cell. In general, there's three arms of ER stress response. One is IRE1 alpha, PERK, and ATF6. These three proteins located on the ER compartment. And then when you have stress response, the IRE1, PERK, and ATF6, they have downstream transcription factor will initiate target gene expression level that control unfolding protein responses or stress challenges. So we know that in the environment, you normally will have protein aggregation or nutrient deprivation or reactive oxygen species or hypoxia. This situation definitely will initiate ER stress response. And this is not like a protected things. They will protect the cell to deal with this harsh environment. And when you have this, they will initiate certain mechanism to maintain the cell proliferation, to maintain the cell survival, and then will eliminate, will cause inflammation, will eliminate inflammation, will also have immune responses. Also in the tumor cells also have this ER stress response, and this will help tumor to fight against stress from the environment, and then they will help them to do immune evasion or metastasis. So this stress response is very diverse and then very interesting in different cells. And then we want to understand whether these tumor social macrophages in the tumor microenvironment. We know tumor microenvironment is very stressful and nutrient deprivation. So how these macrophages can survive, they definitely have some mechanism to control this situation. So we took a public available data set from the mouse peritoneal macrophages stimulated with IL-4 or lung cancer or liver cancer patient tumor social macrophages. As you can see here, this mouse model IL-4 stimulated peritoneal macrophages, we call it M2 macrophages, versus naive. These M2 macrophages have high ER stress response. Similarly, in the tumor social macrophages, in the lung cancer and liver cancer, these tumor social macrophages are enriched with this ER stress response. One of particular pathway we are interested in is PERK. So this couple of pathways have been shown that the importance of the ER stress response, like IRE1 alpha, important for macrophages, and we're interested in the other pathway called PERK. So this PERK is also on the ER compartment. When you have unfolding protein or chronic inflammation, you will initiate PERK activation. So you will see the PERK phosphorylation, and they will upregulate downstream EIF2 alpha phosphorylation to initiate ATF4, and then go into nucleus to initiate downstream genes expression level to cope with this stress response to maintain cell homeostasis. So we want to know whether this pathway is essential to support M2 macrophages. So we simply looking at our previous published data. When the postdoc, we have RNA-seq data. We took this data set, compare M1 macrophages stimulated with ILPS interferon gamma, and then the M2 macrophages stimulated with IL-4. We can see this PERK pathway, this EIF2AK3 is PERK, and also downstream pathway are enriched or increased by these M2 macrophages. Similarly, we took TCGA data set, compare the colon cancer, lung cancer, or PDAC. To do the analysis, we can see these PERK or ATF4 downstream transcription factor are partially correlated with CD68, these human macrophages markers. These data suggest M2 macrophages or tumor social macrophages, they are having increased expression level of the PERK pathway, and suggest then PERK ER stress response is important to these M2 macrophages. So in order to validate these data, we do the experiment. We took bone marrow derived macrophages stimulated with IL-4 compared to M0 and naive macrophages. You can see this possible PERK is increased by M2 compared to naive. And we transplant B16 F10 melanoma to mice and took the spleen macrophages or or tumor social macrophages compare the PERK phosphorylation, can see these tumor social macrophages have strongly induced the phospho-PERK expression level in these tumor social macrophages compared to naive stem macrophages. And then you may argue that how about we induce ES response by drug? So there's a drug called tasigargan. There's a agonist can initiate PERK activation. So as people shown before, tasigargan definitely can increase the PERK phosphorylation, induce the ER stress response. However, we're looking at whether this tasigargan stimulation alone, whether can induce M2 phenotype. We found stress alone cannot induce M2. And this M2 marker, CD206, CD301, you have the tasigargan to induce phospho-PERK, but M2 activation is not happening compared to the IL-4 stimulation. So this data suggests PERK activation is essential to support M2 phenotype, but it requires extra signaling to synchronize together to promote M2 phenotype. If you have a long stress response, a long stress inducer, you cannot initiate the ER stress. So in the tumor microenvironment or IL-2 or IL-4 stimulation, it definitely can initiate M2 phenotype, but tasigargan alone cannot. So in order to prove this concept, we generate conditional knockout. We cross the PERK flox mice to lysine to see whether when we knock out the PERK, can we affect M2 activation. So this is wild type flox and looking at the 206, 301 or PD-L2 Ramapha, after IL-4 stimulation, you can see a robust activation of these M2 markers from wild type. However, when you knock out PERK intrinsically, you decrease this level of activation from 29 to 10, 40 to 30%. So definitely when you intrinsically inhibit or delete the PERK, you will affect M2 activation. Similarly, we co-culture this wild type or knockout mice with B16F10 melanoma cell or lung cancer cell line in vitro. So you can see when you co-culture wild type bone marrow derived macrophages with tumor cells, you can see a robust M2 activation. But when you knock out PERK intrinsically, you see a strongly reduction of M2 activation from this B16 melanoma or lung cancer LLC cell lines. And then we want to see whether this will affect the immunosuppressive function for T cell proliferation. We know that M2 or immunosuppressive macrophages can suppress T cell proliferation. So you can see we use a OT1 T cell. You can see the OT1 T cell can proliferate robustly from about 65%. We co-culture with M1 this pro-inflammatory macrophages. They cannot suppress the T cell proliferation between wild type and knockout. I can see when you have OT1 co-culture wild type M2 macrophage, you can see the proliferation from 65 down to 15. But when you knock out PERK, the suppression is decreased. So T cell have higher ability to proliferate under this knockout M2 macrophages. We quantify here. So this suggests M2 immunosuppressive property requires PERK stress response to maintain the immunosuppressive function and M2 activation. So we always go back to the in vivo study to see whether it's true for the tumor progression. So when we transplant B16 melanoma to wild type and PERK knockout mice, can see wild type mice have robust tumor growth compared to knockout. The knockout mice have low tumor volume after 16 days, and then tumor weight is strongly decreased by intrinsic PERK knockout from macrophages. And then we're looking at the immune phenotype. You can see here, when you knock out PERK, the total number of tumor associated macrophages decreased by day 16. And then M2, CD206 positive macrophages also decreased by knockout. And you have more infiltrated T cell numbers, and you have more gamma CD8 T cells, CD4 T cell from the knockout mice. So these data strongly suggest when you eliminate this PERK expression level in tumor associated macrophages, you will suppress immunosuppressive function. We will increase the T cell infiltration, increase anti-tumor immunity. And then also this conditional knockout also prolong the survival rate compared to wild type. From here, you can see the wild type mice die around 40 days, but these knockout mice survive longer and they have a better survival rate. So now our data suggests the PERK is essential to support immunosuppressive function, and we want to know how PERK control metabolism. Is this critical metabolic hub in M2? So we did a RNA-seq analysis between wild type and knockout M2, and we also did a metabolic analysis to see. So we can see RNA-seq data showing that when you knock out PERK in M2, you have reduction of lysosomal function and amino acid metabolism, carbon metabolism, also mitochondrial acidic phosphorylation. So this data strongly suggests when you knock out PERK, you definitely can intervene the metabolism network in M2 macrophages. And we also did the metabolomics analysis that echo our RNA-seq data showing that we have reduction of amino acid metabolism, lysosomal function, and mitochondrial acidic phosphorylation by this PERK knockout. So we know that M2 or tumor-associated macrophages, they require mitochondrial function. This has been well studied a couple of years ago, and when I postdoc, I showed that lipid metabolism is important for M2 macrophages. So we then looking at the mitochondrial structure, whether this PERK knockout will affect mitochondrial structure. So we can see here, wild type M2 macrophages have beautiful mitochondrial structure, the crystal structures here, and then it's very beautiful, and then very functional. But when you looking at the PERK knockout M2, the mitochondrial become funky, and this crystal structure become wild and loose. And crystal structure is important place for mitochondrial acidic phosphorylation. There are lots of electron transport chain protein located on here. There is a place to do that. When you knock out PERK, it will affect mitochondrial structure functionality. Quantify here. We also check the mitochondrial electron transport chain for the acidic phosphorylation. As you can see here, when you knock out PERK, those genes essential for mitochondrial function are strongly reduced or decreased by knockout. And we RNA-seq data here, and so looking at the protein level, when you're looking at the M2 knockout, this complex 1 to 5 protein level is decreased compared to wild type M2. So the data strongly stress PERK deficiency affect or interrupt the mitochondrial fitness. And also we quantify mitochondrial mass. We know that M2 macrophages require mitochondrial function. They have bigger mitochondrial mass. When you knock out PERK, your mitochondrial mass is decreased, quantified by mitochondrial green. Also, we use the seahorse analysis to see this oxygen consumption rate. By seahorse analysis, you can see these knockout mice have low mitochondrial activity, low oxygen consumption rate, also have low ATP production. So these data strongly suggest if you knock out this PERK stress response pathway, you will interrupt or impair mitochondrial function to support M2 phenotype. So we looking at this analysis, we can see what else is also affected by this PERK knockout in addition to mitochondrial function. Two things we found interesting. One is serine family amino acid metabolism, and the other thing is glutamine metabolism. And that time this is not really clear how this non-essential serine metabolism is important for M2. But from this data we found when you knock out PERK, we have strongly reduction of serine metabolism pathway and also glutamine metabolism pathway. Why is that? So simply introduce what is serine 1-carbon metabolism. Serine is a non-essential amino acid, so it can be synthesized intrinsically. We know that this glycolysis and glutamate metabolism are essential for M2, has been published a while ago, and this can support mitochondrial function running into DCA cycle or mitochondrial function to support M2. And serine metabolism is a branch of glycolysis. So from glucose and to 3-PG, and then this 3-PG can branch out to serine synthesis in the cell. So through PHGDH, PSA1, PSA81, and PSAPH, these three are only three enzymes involved in the serine intrinsic synthesis. And then the serine also can be taken up by from environment. And then the serine can be catabolized to glycine. They support fatty Foley cycle, methionine cycle to do the nucleic acid generation or epigenetic regulation like methylation or DNA histone methylation, or generate NADPH. And this pathway is important for pregnant women because for the brain for brain development is this serine or Foley cycle is important for baby brain development. So it's really important for our body. And it's unclear whether this pathway is essential to support M2. So a couple of years ago, serine 1-carbon metabolism has been suggested important for T cell, CD8 T cell expansion, published in Cell Metabolism in 2016 by Ross D. Jones, and also two papers from Marcia Haggis at Harvard, and then Jeffrey Ruffalo. They're showing serine 1-carbon metabolism pathway is essential to support T cell expansion and immunity, but it's unclear whether the serine metabolism is important for macrophages. So we did simply analysis. We also took publicly available data sets to do the analysis. We found that this M2 up to IL-4 stimulation in mice, we can see this M2 have enriched in serine glycine 1-carbon metabolism compared to naive mice, naive macrophages, and lung cancer macrophages or liver cancer macrophages from patients. These macrophages enriched in serine 1-carbon metabolism, similarly in the liver cancer tumor-associated macrophages. So this data strongly suggests serine metabolism may play an important role to support this M2 or immunosuppressive tumor-associated macrophages. So we took our data set, compare M0 and naive, M1, M2. We found the serine 1-carbon metabolism pathway is enriched or elevated by M2 compared to M1 or M0. And then we also did the TCGA data set analysis. We took the CRC tissue, compare these three key enzyme, PHDH, PSP1 or PSPH, serine synthesis enzyme, to do a correlation with human CD68 macrophages, and see these three enzymes are partially correlated with the human macrophages CD68 expression level. So this data strongly suggests serine metabolism may be important for M2 or tumor-associated macrophages. So next, we did a metabolic analysis, compare M0, M1, M2 to see this metabolite level from these macrophages. As you can see, 3-PEG is a branch out metabolite, is high in M2. Serine level is high in M2 compared to M1. Also downstream glycine also enriched in M2 macrophages compared to M1. So we want to know whether this pathway or the expression level is controlled by PERK, because we saw PERK knockout, we have a reduction of serine metabolism. So we want to know whether PERK control these three gene expression level. So we took our RNA-seq data and see knockout M2 macrophage have low PHDH, PSA1, and PSPH expression level. And then we next measure the serine concentration from M2 macrophages between wild type and knockout. And see this PERK knockout, you have low serine production compared to wild type. Later, we want to know, it has been shown a while ago, showing that PERK can control ATF4 expression level, and ATF4 can control the three gene expression level. But we want to know whether it's the scenario happens in macrophages. So we also took a public available data set to do analysis, compare M0 and M2, and this ChIP-seq analysis looking at the ATF4 ChIP-seq. You can see ATF4 bind to certain genes important for M2 mitochondrial function, like here, and also serine metabolism. So we can see this ATF4 involved into the PSA1. They bind to the promoter of PSA1. So suggests the ATF4 definitely can control the PSA1 expression level to promote or sustain the M2 phenotype. And then long story short, we generate, we also use our knockout mice, PERK knockout mice, to check whether PERK knockout have reduction of ATF4. You can see here, PERK knockout, we have low ATF4 expression level, low PHGDH, low PCL1 compared to wild type. And then we looking at the ATF4 knockdown. We want to know whether ATF4 definitely can affect the expression level of the serine synthesis. You can see when you knock down the ATF4 by hairpin, you can see PHDH expression level decrease by ATF4 knockdown. Also, PCL1 also decrease. So suggests you knock down ATF4, it definitely can interfere or impair this gene expression level essential for serine metabolism. So we knock down the ATF4, looking at the expression level of M2, you can see wild type and non-wild type. The control luciferase hairpin had robust M2 activation, but when you knock down ATF4, you can see this expression level M2 is decreased. So suggests ATF4 is also controlled by PERK involved in the M2 activation. So next, we want to make sure PCL1 or serine metabolism is essential. So we next generate another conditional knockout. So we generate PCL1 flox mice cross to a lysine cree. You can see here is a flox mice wild type. You have robust M2 activation, 206, 301, and PDL2 ROM alpha. But when you intrinsically knock out PCL1 in macrophages, you can see a robust reduction of M2 activation in 206, 301, and PDL2 and ROM alpha from 64 to 25. So we also did other assay, like use hairpin to knock down PHGDH, PCL1, also use small molecule showing the same thing. So when we interfere this pathway, we definitely can impair the M2 activation. We always go back to the vivo, so we transplant B16F10 melanoma transplant to the PCL1 box wild type mice or conditional knockout. You can see when you have wild type mice, the tumor robustly grow after 15 days. But when you conditionally knock out PCL1 in macrophages, you can see a strong reduction of tumor volume and tumor weight strongly reduced. And similarly to PERK knockout, we can see PCL1 conditional knockout have low total number of tumor-associated macrophages compared to wild type, low M2 CD206 positive tumor-associated macrophages compared to wild type, and you have interesting induced the infiltrated T cell TL numbers increase. You have induction of the interferon gamma positive CD8 T cell and CD4 T cell infiltrated to the knockout mice to eliminate tumor progression. So far it's great. And we were very ambitious to see what else that PCL1 can control. And then, so we know that PCL1 and PERK are important for M2 elevation, and they're important to support serine metabolism. And then we know, when we noticed that the serine synthesis pathway require glutamate. So previously we found the metabolism of glutamate metabolism decrease when you knock out PERK. And then this pathway PCL1, when you have PCL1 to generate serine, we require glutamate to donate nitrogen to this pathway, and then to generate serine. And this byproduct will be alpha Kg. And we want to see when we perturb this pathway, does that affect or do that affect the alpha Kg production? If that's true, they definitely control this pathway. So we're looking at the PCL1 knockouts. We can see when you knock out PCL1, this alpha Kg production, alpha ketoglutarate production is decreased compared to wild type. Similarly, when you knock out PERK, you have reduction of the alpha Kg generation. So the data strongly suggests serine synthesis is important, and we also require this nitrogen to achieve this process. And the byproduct alpha Kg is affected by this interference. Importantly, we know that from a while ago, alpha Kg is important cofactor for JMJD3, and JMJD3 is known that histone demethylation factor contribute to epigenetic regulation to support M2 phenotype. So we wonder whether this pathway also contributes to this GNJD3 activity to support epigenetic modification. So we looking at the PCA1 knockout M2 versus wild type. You can see PCA1 knockout, we have the PCA1 protein level decrease, but you see hyper methylation of H3K27ME3 present in knockout. So no more demethylation. You have hyper methylation for knockout. Similarly, the PERK knockout shows the same thing. You have hyper H3K27ME3 histone methylation from these two knockout mice and two macrophages. And then you may argue this probably affect the GNJD3 gene expression level. But obviously, when we knock out these two genes, PERK or PCA1, the GNJD3 gene expression level is comparable. We also have a protein level, they are also comparable. So this knockout not affecting GNJD3 expression level, it may affect this cofactor for this activity. So when we have this data, we submit to Nature Immunology, and then the reviewers or editor ask, this is a bulk. You should have a specific chip-seq to show that. So during this revision, we did another chip-seq. It costs a fortune to do that, but anyway, as long as we can get into Nature Immunology, we'll do it. So we did. So we looking at the PERK knockout. So when you knock out the PERK and see these M2 genes, IRA4, PPAR gamma, PHGDH, MGO2, the CD301, they have hyper methylation from this H3K27ME3. And this phenotype not affecting M1 genes. So M1 genes is comparable between wild type and knockouts. So this knockout definitely affect hypermethylation only specific to the M2 genes, IRA4, for instance, or PPAR gamma or PHGDH. So the last experiment for this, we do the rescue experiment to see whether it's true or not. So this data is kind of very busy, so I'll go through with you as one by one. So the black bar is wild type. And we know when we knock out the PERK or PCA1, you will have reduction of alpha Kg. So in this situation, if we artificially supplement alpha Kg or DMN Kg, can we rescue? Because alpha Kg is essential for GNJD3. So that may be rescued. And then the next condition is we supplement alpha Kg to increase activity of GNJD3, and then we put selectively inhibitor of GNJD3, GSKJ4, that will reverse. So the two condition knockout show the same thing. And looking at the M2 genes, so you can see knockout the PERK or PCA1, you have reduction of expression level of M2 genes, but when you supplement alpha Kg artificially, you slightly increase gene expression level from M2. And then, so that suggests alpha Kg is essential, and that will promote GNJD3 activity to do a DNA histone demethylation to support M2 gene expression level. And then next, we suppress GNJD3 activity by GSK when you have Kg. You can see reverse, everything reverse. So this data suggests this stress response controls serum metabolism to generate alpha Kg. This alpha Kg generation is essential to asset the GNJD3 activity to support gene expression level essential for M2. And when you perturb this pathway, you will decrease this scenario. So we always go to the immunotherapy or gene cancer therapy. So what we did for this experiment, we used the PERK inhibitor or PHGH inhibitor to intervene the serum synthesis in the mice. You can see wild type mice, normal mice with vehicle tumor grow robustly, but when you have PERK inhibitor administrate IP, you can see reduction of the tumor growth, and then also inhibit the serum metabolism. You will decrease the tumor growth. Tumor weight also decrease. The total number of tumor social macrophages also suppressed by PERK inhibitor or serum synthesis inhibitor, and reduction of the M2 phenotype. And we found this the PERK inhibitor increase the TIL numbers, increase the interferon gamma CD8 T cell, interferon gamma CD4 T cell, but the serum metabolism inhibitor decrease T cell. So remember, previous data already show serum intrinsically is important for T cell expansion. So there might be side effects that inhibitor affect T cell expansion to the TIL, to the tumor microenvironment. And even you can see the reduction of tumor size, but the T cell induction is not really significant. But the PERK inhibition definitely induce the TIL and then prolong the survival rate. So based on this, we took this PERK inhibitor combined with PD-1 to see whether we can enhance the efficacy of immunotherapy. So the last experiment, this is the B16 melanoma treated with different inhibitor or antibody. So it is a control with IgG2A control, the tumor robustly growth. But when you have anti-PD-1, tumor size decrease to 2 out of 10, under the 1,000 size. But when your PERK inhibitor strongly reduce the tumor size, but you combine these two treatment together, you can see a remarkable reduction of tumor growth from this treatment. So this data strongly suggests when we perturb the stress response in the tumor-associated macrophages, you will reduce serum metabolism, you will increase the anti-tumor immunity by increase the infiltrated T cell, produce interferon gamma to kill tumor. Furthermore, when you combine this immunotherapy treatment anti-PD-1, you will further promote efficacy of the immunotherapy. So in summary, we found that in the tumor microenvironment, these tumor-associated macrophages or immunosuppressive macrophages require this stress response. The tumor increase the stress response, increase the PERK phosphorylation, and its PERK activity sustain the support mitochondrial activity. That reason macrophages, tumor-associated macrophages, they have intact mitochondrial function to maintain their functionality. And then this PERK promote mitochondrial function, and this PERK also upregulate downstream transcription factor ATF4 that will promote expression level of PHGDH, PCL1, and PSPH for serine synthesis. And this serine synthesis is essential to generate serine to support metabolism. And this byproduct, alpha Kg, can be a cofactor that will activate the activity of JAM33 to support the epigenetic modification that is essential for supporting M2 phenotype. And then we still don't understand what else serine can do to the immune suppressive macrophages. So currently we are working on how the serine contribute to this mitochondrial activity, how the serine converted to folate cycle, that will convert it to methylation cycle to do the epigenetic regulation. So it's the further project we are working on currently, and then the student project we try to understand how serine contribute to mitochondrial activity, and then it's an ongoing project. And then the data I show you today is already published in last year 2022 in Nature Immunology. And if you are interested in the details, you are welcome to take it out to have a look. And lastly, I would like to thank my group. I have a very small group, so this project is leading by my PhD student Lydia Rens, and she's going to graduate next year, and she got a postdoc position at Harvard. But anyway, I really appreciate my group, and they work very hard. And we are a small group, but we always aim big, and we try our best to address our questions and achieve our dream. And also thanks our collaborator from CASE, my previous institute, Alex Huang, to support resources. Stanley give us the PERK knockout, PERK flux of mice, and other people for the machine and then analysis. Also, my collaborator from Lausanne, University of Lausanne in Switzerland, Pinchi Ho, one of the leader in the immunometabolism T cell, So he helped me to do the metabolomics analysis in the institute. And also thanks another collaborator from Ohio State, Jerry Liao, to help us to do the epigenetic ChIP-seq analysis. And also thanks for the funding agency, American Cancer Society, CRI, and then my previous institute case, and then the MRF Melanoma Foundation. And thanks for your attention. And then any question you can ask me. Thanks.